point about pipeline means that these are therapeutic approaches that are in development, so things to come for the future. And some of these are close, and some of these are long-range objectives. So the first approach would be to use a small molecule, a drug, to somehow better manage symptoms. We all know that we've heard today that although there are approaches that can be used right now, clearly the level of control is lacking, and there's plenty of room for more improvement. And the first approach would be, why don't we just go after the problem? Why don't we go after the mutant channel with some kind of small molecule or drug that can somehow fix the problem? And there's a clear opportunity to do that in the case of hypokalemic periodic paralysis. So this cylinder represents a muscle fiber, and each of those circles are the different channels and transporters that are there. And the one in red is the one that's defective in hypo-PP, which may either be a sodium channel or a calcium channel. And what's really astounding and helped us and others unlock the key and understand what's wrong in hypo-PP was understanding the details of these mutations. We haven't done this yet this year, but each one of those cartoons on the left and the right is one way of looking at an ion channel. The gray bar represents the membrane, so the outside of the cell and the inside of the cell, the membrane separates them. And that long string would be a string of pearls. That's the different amino acids that make up the protein that's coded by the gene. So we can use this to understand structural parts of the channel and where are these variants that are detected that you hear about in your in vitro report. So each of those little red triangles is a variant that was found in a family with hypo-PP. And the amazing thing is they're all in a similar homologous area. It's always in the little cylinder with the pluses. And all of those variants, except for one, begin with the letter R. And that's the shorthand that those are all at arginine, a certain type of amino acid, in the voltage sensor of the channel. There is no way that is by chance. So this is telling us something about what goes wrong in hypo-PP with the channel. And to make a long story short, the answer is these mutant channels are leaky. So this is a different cartoon view of a channel. Again, the membrane is that horizontal stripe. The channel is a little potato in there with the pathway for the ions to move through. And the red cylinder is the part of the channel that's sensitive to membrane voltage, because these channels open and close in response to change of the voltage. That's how the whole system works, to send a signal when it's time to contract. And so there's supposed to be one area that moves in response to voltage and a different part where the ion channel comes through the pore. What happens in hypo-PP is each one of these mutations is in that voltage sensor segment. And it makes that little red cylinder not fit correctly so that there's a leak. There's an inappropriate pathway for sodium to come leaking into the cell. And that's the problem. They're leaky channels. And all of the mutations are the same because it creates the same type of leak. Well, that's a great target, right, for therapeutics. Let's plug the leak. Everything should get better. It turns out that this is an unusual part of the channel. There are no pre-existing drugs or medications, because all of those drugs that act on ion channels, things like mexilotine that you've heard about, they act on the conventional pore. That's a different part. This is a leak that's next door. So with some colleagues at the University of California in Irvine, they're studying an unusual channel that has a leak through the cylinders of the channel. That's what this molecular structure is showing. And they identified a lead compound that can potentially block the leak. And we tried it in our mouse with hypo-PP. So this is the calcium channel R528H mouse. The graph is showing, again, we're measuring muscle force. That's a little cartoon at the bottom right. We can take a muscle out of this mouse, give it a potassium challenge, and see how it deals with that stress. So again, the vertical axis at the bottom is force over 90 minutes, and you can see everything is very stable. We're measuring the maximal force every 2 minutes, relative value of 1.0. Then the potassium gets decreased down to 2, and the untreated muscle in the black symbols, there's a big 50% loss of force. So that's hypo-PP in a mouse model. And when we put this small molecule on, this lead compound, that's the blue trace, and you can see we prevent about 2/3 of the loss of force. So there's a ways to go, but this is the first and only novel molecule that's been identified that potentially blocks this pore. So the beautiful thing is we have a new grant and a partnership with some computational chemists at UC Irvine. We're going to get atomic crystal structures of mutant channels and do docking studies to design and make custom molecules to better block the pore. So that has tremendous potential. I'm not going to spend time at all on this. Just everybody says, well, you've got this idea. When can I go to the pharmacy and pick it up, right? So there are a lot of steps. Just to say there are a lot of things we have to do, okay? So that was having a drug that would act on the misbehaving channel. And in fact, when ion channel diseases were first discovered, and muscle, periodic paralysis was the first one, the pharmaceutical industry was interested, let's find small molecules that can make the misbehaving mutant channel act like a normal channel. And that approach only got so far. This will be a new chapter in that approach. Another technique is to say, look, we understand the consequences of having one of these misbehaving mutant channels, which is to have the battery run down and lose the excitability of our muscle fiber. So let's take another approach and have a drug that's acting on a different channel, on a normal channel, but have a way that we can supercharge our battery and prevent it from being susceptible to the untoward effects of a mutant channel. So to remind you, we've looked at this a couple of times today. Now on the vertical axis, you're looking at voltage versus time really fast. Remember, the problem that was happening, is that compared to when you are normal, the panel on the left, and your battery is charged, that wiggly trace is down low. During an episode of periodic paralysis of any flavor, actually, your battery is partially run down. That resting potential is moved up, and that renders the fiber inexcitable. So it turns out we know from many years of experience of working with excitable cells that if you could open potassium channels in the muscle, open a channel that allows potassium to go in and out of the cell, that would help fortify this battery and make things more stable and could potentially prevent an attack of paralysis. This is how we came upon trying retigabine that I told you about 2 years ago, but there's a little bit more to the story. So first, here's the electrical demonstration. Again, this is a recording from one of our hypo-PP mice. In the bottom is now that voltage. How well is your battery charged over 140 minutes? And what happens, that black trace there, everything is fine and stable, and then we lower the potassium. That's the box on the top. When we lower the potassium, that's the stress. That trace starts to go up. That means your battery's running down. So that's showing, yep, this mouse is susceptible to hypo-PP. But if we add this drug retigabine, which opens a potassium channel, a special kind of potassium channel, and repeat it again, you see how the battery stayed charged. We prevented that rundown by compensating with by activating a different channel. That's what it looks like in voltage. This is what it looks like in force. So here we're going back to the most common cause of hypo-PP, the calcium channel R528H mouse. We take the muscle out. Again, we're monitoring the force every 2 minutes. We contract the muscle and see how well it can operate. Everything is very stable in the beginning. We lower the potassium from 4.7 to 2, and the force starts to drop. There's an attack of hypo-PP in our mouse model. Now, the one in red, these are two traces from the two different muscles of the same mouse. Everything's the same, but then for the symbols in red, we add retigabine to the dish, and the force comes back. The important thing is not only can opening these potassium channels with retigabine help stabilize the resting potential, they can rescue the situation if an attack has already started. So this has potential, therefore, to be used as a rescue therapy after an abortive therapy after the attack has already started. If you apply the retigabine before the potassium channel, that's the one on the left, it's a different mouse, so the attack is a little different. It's those wiggly black lines. But you can see the potency of this approach. If the retigabine is provided in a preventive aspect before the low potassium challenge, there is no loss of force whatsoever. So this has tremendous potential. This is the dose response. The bottom is the concentration of the drug. And on the y-axis, you're looking at how much protection you're getting. So the bigger the number, the more protected you are. And the point is, for either sodium channel or calcium channel hypo-PP, either one of those mice, it's really impactful level of protection, even with as low as 1 micromolar concentration of the drug. This is something achievable in the body easily, as opposed to the black curve. Years ago, Frank Lehman-Horn and others, with what was available at the time, tried a different potassium channel opener, panacetyl, and you need 10 times as much to get the effect, and you couldn't use it because at that high dose, it would reduce your blood pressure and you'd be at risk of fainting. So we have a more potent drug with a different approach that shows great promise. And it also works in hyper-PP. So this is our hyper-PP mouse. We just did this last week. So the attack is a little odd in this one example. It starts late. That's the open black symbols. And if you pre-treat with retigabine, you can provide a level of protection. So we're really excited that this class of potassium channel openers has potential both to be preventive and abortive therapy for both hypo-PP and hyper-PP, as well as, this is not the new version of the slide. I can just tell you it also works for Anderson-to-Will syndrome. So we have a pharmacologic model of Anderson-to-Will syndrome, and this provides protection from that as well. So when many of you heard this story 2 years ago, it's like, you know, retigabine, which was marketed initially as an anti-epileptic drug, where can I get it? It's taken off the market because of adverse effects, some stressful for the liver, very mild cognitive effects, but the big thing was there was a discoloration, blue discoloration of the skin and the whites of the eye, concerns that it would affect vision, and so it was pulled from the market. And there have been continued interest in this drug. It was a whole new class of drug to be used for epilepsy and other neurologic conditions. And so another company has bought up the rights to retigabine and has modified it. They're now in phase 3 clinical trials for epilepsy. And the adverse effect profile is much more favorable. And so I think pretty soon there's going to be an opportunity for off-label use of this new derivative of retigabine that hopefully will be out and FDA approved pretty soon. It's in phase 3 trials. So this is mentioning what we'd like to do here. From the periodic paralysis scope, we want to make sure we objectively assess and confirm effectiveness in patients. So we can do things like look at, on a short time scale, the ability to protect the long exercise test of the compound muscle action potential, get a quick answer as to whether there's any benefit, and then there'll need to be a well-controlled, random controlled double crossover trial to really show effectiveness, and then we'll be able to get approval, I'm sure, for this class of medication. The other approach that I began to mention last year, and we've made a lot of new progress that I want to speak to you about, is to use gene therapy. So, of course, this could be applied to any inherited disorder. There are a couple of reasons why periodic paralysis is a particularly attractive and good target for this approach. One, we know in classical cases it's a single gene that's affected, and we know what's wrong with the mutant channel. The other is that both from work in animal studies and in case studies of patients, we know that the problem is, the mutant channel misbehaves. So it's not as though it's missing or absent, it's behaving in a promiscuous, abnormal way, and that's what's causing the susceptibility to periodic paralysis. Well, we know that if you just got rid of that bad copy, you have 2 copies of each gene, if you got rid of one and only had one normal, that is sufficient in humans to have completely normal function of skeletal muscle. In fact, there have been incidental findings, people doing genome-wide screens for other diseases where the individual had no muscle symptoms whatsoever, and one copy of the sodium or the calcium channel gene was completely disrupted, and they're asymptomatic. So that tells us we can use that approach. So the first approach would be, let's get rid of the bad copy. And so the molecular tool there that's out there in the press you've heard about is CRISPR. CRISPR is basically a molecular technique where you can precisely write the zip code, write the address for where to take your scissors and go in there and make a cut in the DNA. And this can be done inside cells, not just in the dish. That's the amazing, beautiful thing about CRISPR. The idea is you would start with the scenario on the left side, which is our model of a muscle fiber that unfortunately in the bottom right in the red circle has the misbehaving channels with a missense mutation. You could use CRISPR to selectively disrupt only the mutant channel, not the wild type, and then that should hopefully ameliorate the symptoms. We've made substantial progress with this. You start by trying to make these manipulations in the dish. It's technically easier, so we isolate muscle from a hypo-PP mouse. CRISPR is that circle. It's encoded by a DNA plasmid. You get that to be taken up into cells. The cells that successfully took up your machinery, you tag the system so that those cells will shine green. You can pick those out and then look and see what you did to the DNA. And technical details, just to show you how clear it is. The case on the left, you put these tools into a wild-type muscle fiber, and there were no accidental cuts with the scissors. What these red bars show is what percent of the DNA is entirely normal with no insertions or deletions. That's the center bar in the middle on the left there. And so we didn't make any accidental cuts in the normal copy of the gene, essential for this to work. But on the other side there, when we applied this to the R528 muscle from that mouse, you can see all those other little bars. That indicates we chopped out 1 base, 2 base, 3 bases, so we scrambled it. And that disrupts that gene so it can't make a functional channel. So we show in the dish that we accomplished that. The next big thing is do it in the animal. And so there's a way that we can use to get DNA to be taken up by muscle fibers in the foot of a live mouse, allow a couple of weeks for that machinery to do its work, to modify the gene, for the ion channels to turn over, and then go back and we can measure how well the muscle functions. On the bottom, what that's just showing on the far left, under bright light, that's the little hand of the hind paw of a mouse showing the muscles. And you can see they're nice and bright and green because we've successfully introduced our molecular machinery to do this gene editing. And you can isolate single cells as shown in the bottom panel on the right, where it looks like little green rods. We've isolated those. You can then pick them up and do the DNA test. And again, what we're seeing now is not in a culture dish, but in a mouse, in the foot of a live mouse. We were able to disrupt about 50% of the mutant copy of the gene without deleteriously accidentally cutting the normal copy. So this is where I was with you 2 years ago. And with the support from the PPA, we were able to generate additional preliminary data to now see what does this mean for muscle force. Because we were electrophysiologists and muscle biologists with no track record in gene therapy. So to get a major grant from the federal government, you need a track record. So it's a chicken and the egg, how do you get started? And the support from the PPA was critical. And with that money, I'll show you what we were able to do. And we got 2 grants this year because of this preliminary data. So here's the first one. We have this hypo-PP mouse. We introduced our molecular tools with CRISPR to destroy the mutant, the bad copy, the R528H copy. And what you're seeing here, each of these traces is force, muscle force in the vertical scale, and time. And what you're looking at is a twitch. The muscle contracts and relaxes because we've stimulated it. And we're stimulating it maximally in normal potassium. And what you see in black on the left, sham means that's the other foot from the hypo-PP mouse where we didn't do this therapy. And it can still generate some force in normal K. But oh, look at the one where the CRISPR was. The force is a little bigger. Look what happens when you put in low potassium. You get an attack of weakness in the untreated foot of this mouse, but the very same mouse from the other side doesn't get an attack of hypo-PP at all. And when you look at many, many mice, let's look first at the left. That's the baseline force before any potassium challenge. So in black, that's scatter. That's a wild-type mouse. In red, that cluster of points where there's about a 30% baseline lower force, that's our hypo-PP R528H mouse. So the mouse runs around, it eats, it makes little mice, does everything it needs to do, but the muscle force isn't quite at the same level of performance. In blue are other mice from this same colony where we did the CRISPR deletion of the bad copy of the gene, and you can see the baseline force is much better. What's even more impressive, on the right here, now we're doing the potassium challenge. So again, looking at relative force on the vertical axis and time over 70 minutes there. So every 2 minutes, we do maximum contraction. How well is this muscle working? And we normalize everything to 1 at the beginning, and then do the low potassium challenge there. See, we drop down to 2 millimolar. And all of these are paired measurements. 1 foot untreated, the other foot from the very same mouse treated. All the red ones are untreated. There's about 30% loss of force in this test. We're so proud of this. We show every single example, not even the averages of all the blue ones. Every single mouse that we introduced these molecular tools was cured. Does not have hypo-PP because of this. So we're really excited about this, as you can imagine. There's still a lot of work to be done, but this shows tremendous promise. There's another form of gene therapy, which is not to ablate the bad copy, which again, only because of the biology of hypo-PP, we have a chance to even do that in the first place. But there's another tool. The CRISPR tools have been adapted and modified. They're always improving. So now you can actually make a correction. You can replace. So in every case of periodic paralysis, the mutation is a single nucleotide that's the wrong one, the wrong piece put in the instruction. So you can take out the wrong one and put back a normal one, which is the other approach that we're now trying. So this has only been done so far in the dish. And I know these are kind of removed esoteric results, but again, I want to show the raw data because we're proud of it. So what you're seeing in these funny humps, this is the data that you get back when you do DNA sequencing. So there are 4 different channels, 4 different colors. This is your A, C, T, G of the genetic code. So we start there with the R528H mouse on the left. And as you go along where you see that A to G, there's an A there, which should be a G. And when we did gene editing, the efficiency is not quite where we want it to be there. But you can see, it's hard for me to look from this direction, we modified about 34% of the bad copies were turned into normal copies, which is really terrific, because we know that we're not going to need to correct every single copy. We know that because for many of you, your muscle performance is fine most of the time. It's these intermittent attacks that disrupt your life. And so you're right on the knife edge of, just bad enough that you're susceptible to these temporary attacks, but not so bad that you have weakness all the time. And the mathematical modeling and some of our drug models with the animals say you probably only have to correct maybe 30 or 40% of the mutant channel, and it's going to be enough to take you over the hump and remove the susceptibility to attacks of weakness. The other example here, it's another DNA sequencing run. This is from the hyper-PP mouse. So we're doing hyper-PP as well as hypo-PP, and we were able to change 26% of the G back to A, which corrects for the hyper-P mutation. So we're very excited about this. Everybody's always asking, not only for periodic paralysis, but every disease, how close is this to being available for patients? So there's a lot of stuff we need to do where we need to show that this works not just for the R528H, but for a lot of different other specific mutations that cause periodic paralysis. We need to do more sophisticated screening to make sure there weren't any off-target cuts accidentally. And then one of the things we want to do is use these same molecular tools, but apply them in human cells. Because the mouse genome is very similar to the human, but it's not identical. And the way these little molecular scissors and gene editors work are very context sensitive, the way the DNA is folded and what's next to it. So we may be looking for folks who would be happy to give a skin biopsy so we can grow your skin cells and see if we can correct your mutation in the laboratory, which would be one of the next steps to do for this. So there's the deleting the bad copy of the gene and not destroying the good copy. Similarly, for the gene editing, just to let you know where we are, We have low efficiency, but at least specificity of being able to do this in the dish. We're still working on optimizing the tools to get it to work in the mouse and to demonstrate in the mouse that we've cured the susceptibility to periodic paralysis, but that's what the new money's for. We'll get there. And we will also apply this gene-correcting technology and see how that works in skin cells that we're able to obtain from patients with periodic paralysis. So I'll just finish up by saying there are a lot of challenges in gene therapy for big pharma, okay? So you've seen there are a couple of examples, some rare forms of neuropathy, some types of blood cancer, some diseases in the inner eye. And what the big challenge is the delivery of these tools. So these tools are encoded either by DNA or RNA. And so how are you going to get it to go there? Well, the way viruses work, viruses are beautiful at sneaking foreign DNA into cells. That's how viral infections happen. And so people have taken viruses, modified them so they won't be harmful, and then you can use those as a Trojan horse to bring the DNA into cells. That's worked, and that works really well for diseases of the liver and the spleen. They take up DNA very readily. But it's a little bit of a challenge in skeletal muscle because the volume of skeletal muscle is huge. It's everywhere, all over the body. We have to get every muscle cell. So there are people using AAV delivery models for things like Duchenne muscular dystrophy and FSHD and other muscle diseases. And we will piggyback onto those technologies and try to develop a system that this can be systemically delivered to the mice and show that it can mitigate the disorder and remove the susceptibility to periodic paralysis. There are other techniques that don't rely on viruses. So you can use lipid nanoparticles, essentially putting it in a little oil droplet that gets specifically modified to target where it should be taken up. In fact, this is how your mRNA virus for COVID was delivered to you when you got your injection. So that was eaten up by macrophages, and that's how that worked. So the lipid nanoparticles, there's some work where they're starting to make some headway into delivery to skeletal muscle, but there's still a long way to go. And then the last thing I want to mention is that these tools work well. They can be specific, but you have to think about once you deliver them to the cell, how long are they going to hang around? Because you want them to be long enough to do the gene editing, but the longer they're there, the greater the chance some deleterious thing will happen, that you'll eventually develop immunity against it, it may have autoimmune reaction, or you'll accidentally begin to make inappropriate edits and cuts. So there are new technologies that are being developed to literally make self-destructing gene editing tools. They go in, they do their job, and then they self-destruct. So this is just to sort of tell you what's out there, what everybody is thinking about, why there's still a significant way to go. But what we're going to be able to test with these mice is we can prevent susceptibility to hypo-PP or hyper-PP triggered by potassium challenges. The reasonable presumption is if we succeeded in that and we've corrected the molecular defect, that we will remove the risk of permanent muscle weakness, which is what everybody in this room wants. And if you think about it, gene therapy with a durable therapy is really the only practical way to do this. Because otherwise, a randomized controlled trial, even promising drugs to prevent the late permanent muscle weakness, you might need a trial that lasts 10, 20 years just to begin to answer the first question. Not only is that complex and expensive, but the compliance and to get someone to stick with it for all, it's going to be really hard. But you can do that with gene therapy because you go in and you correct it and it's a permanent change. So we're really excited about that. I always just like to finish on the crew who's helping us. Fen was in the video that opened the conference working with the muscle cells. Actually, she was doing CRISPR experiments then. Marbella does all of our amazing work with the mice. And Marino is a technical whiz that helps with all these measurements. And we appreciate the support. We have new grants coming in from the NIH and the Muscular Dystrophy Association. And all of that was jump-started because of support from the Periodic Paralysis Association. So we thank you for that opportunity. And
And with that, we have some questions for Dr. Cannon. Would you like to come up?
You dropped a card, by the way.
Oh, well.
Professor, does gene therapy, so that we were just talking about, so let's say you cure me, well, let's say if I was younger, and then I had a child, would that child be affected by my gene therapy?
Yeah. Great question. And you can design things either way. So the design and what we're working on now is to target the muscle cells and not the eggs and sperm. So unfortunately, that could persist. All of these delivery systems, you optimize to go to a certain target of interest, but they go a little bit other places as well. So there's a possibility, but the work that's being done now would be used in the context someone who's young, healthy still, doesn't have changes that would lead to the permanent muscle damage, undergo this treatment. It should be hopefully persist for life. But unfortunately, we would need to see whether by chance there was editing in the germ cells that lead to next generations. That would be a different approach or strategy to want to, because what a great thing to have no future generations in your family be at risk. But it would be a
Either way, the child, at least, if it didn't work that as well as that, they would still have a treatment once they're born. And they go through gene therapy, and it'd probably be, by then, you guys would be more advanced.
Exactly.
Yes, thank you.
Sure.
Other questions for Dr. Cannon, specifically on the genetics? Okay, and it's quiet at the internet too. I think a lot of folks still have the kind of rapid fire questions that we've been doing all day. I have noticed, and I'll turn our audience's attention to the fact that we're going to do 2 Q&A sessions tomorrow. So we'll be able to pick up a lot of that too.
Absolutely, a lot to digest. I'll be here all the way through the end of the conference. So.
All right, do you mind if we ask then some questions that would help get that ball rolling while we have a couple of minutes? Okay. All right, so this one comes in. I'm hypo with an allergy to sulfa and tried Keveyis, Diamox, and a few others over the years. Can't remember all I've taken. What would you recommend to talk to my doctor about trying?
Yeah. So as you heard from Dr. Jaqueline, there's a chemical similarity between the carbonic anhydrase inhibitors and sulfa drugs. And so there's an increased risk of rash and allergic reaction. I think it's something to be aware of. It's not an absolute contraindication. So if this individual has never tried those carbonic anhydrase inhibitors, it could be tried. If the response is spectacular from a periodic paralysis perspective, but there are other sort of allergic manifestations, you can even work with an allergist to try to get tolerance. So there are other ways to do it. Otherwise, you're looking at INSPRA and other approaches to minimize loss of potassium in the urine and stabilize potassium level and use potassium supplements right now. The potassium channel openers that I talked about could be a year or 2, and that would be another alternative.
Very exciting. My nephrologist mentioned that you run a specialized test at your lab about PP. What is that test? What does it look for?
Nephrologist or neurologist?
This says nephrologist. My nephrologist mentioned. This is anonymous, so I'm not sure who.
Okay, so
at 4 o'clock.
Interestingly, with primary periodic paralysis, there is no problem with maintaining potassium levels or the way the kidney handles potassium. You can have these fluctuations, and again, they're occurring because the potassium's inappropriately moving into your muscle more than usual during an attack of hypo-PP or out of the muscle during hyper-PP. Abnormalities in potassium may cause you to be referred to a nephrologist. They're the experts on the electrolytes and the balance. But usually a nephrologist's main overlap is the secondary cases that we talked about before, where somebody might have renal tubular acidosis or Gittelman syndrome or Barter's or one of these other where, yes, it's a problem with the kidney, and secondarily, because of extraordinarily abnormal electrolytes, there's weakness of the muscle.
Okay. Do you run special tests at your lab?
No,
That's all right.
I'm here.
Yep, no problem. Does have there been more mutations found? I think we hear this a lot, right? Have there been more mutations found? I'm clinical ATS hypo.
So a great question and always an interesting topic. I think right now the consensus thought is from the experts across the world that for periodic paralysis in general, about 20% of individuals who have clinically convincing support for the diagnosis do not have a recognized pathogenic variant in the 3 major genes. And this issue is a little bit more challenging in Anderson-Tawil syndrome than it is in the other forms of periodic paralysis. I think there's a biological reason for that, which is that in Anderson-Tawil syndrome, you're missing a potassium channel, okay? And there are a lot of genetic ways to destroy a gene or not have it make the product go to the right place. And so it's harder to screen for variants that would be disease-causing, whereas in hyper-PP and hypo-PP, they are missense point mutations in the coding region of the gene. So Anderson-Tawil, it could be in those regulatory regions of the genes. Hundreds of thousands of nucleotides, promoters, all kinds of stuff. It's much more difficult to tease apart. So there are going to be some challenges. Yes, people are continuing to look. I'm working with the Rare Disease Clinical Network at UCLA so that families that have a very compelling story for periodic paralysis but have had nothing identified on conventional screening of the candidate genes, we're trying to look at other levels, going to the RNA levels and everything else. So we haven't given up, and the presumption is there are probably additional genes out there. Maybe they're not channels. Maybe they code for proteins that interact with and modify channels. That's why it ends up looking like a channel disease, but the mutation isn't in the channel itself. It's in an important interacting protein.
Thank you very much for that. I think that's a lot of people's interest. Can you comment on your paper that suggests that the permanent muscle weakness and calcium channel mutations may be due to impaired calcium release and what implications that might have for permanent muscle weakness treatment?
Sure. So it's okay, the slide's down. There's the calcium channel that's involved in hypo-PP functions as the sensor that tells the muscle cell when to release internal calcium. The sodium channel is the one that drives the big electrical wave. The calcium channel is actually signaling when to release. So interestingly, mutations in either channel could cause hypo-PP. And if you look at the patient data, while every form of periodic paralysis has had associated permanent muscle weakness, that's happening, but it's a little bit higher in the calcium channel mutations than the sodium. And so we wondered, maybe it's more than just the instability of the electrical excitability of the fiber, because this calcium channel is doing an additional job.
Would you like me to bring up a slide?
Well, that's okay. That's okay. So the calcium channel is doing a different job, which is releasing calcium. So the paper they're referring to is we measured in our genetically engineered hypo-PP mice, is there a problem not with the electrical status of the fiber, but with release of calcium? And the sodium channel mutations that cause hypo-PP do not impair the calcium release at all. The R528H, it's the only one we had available to test, which is the most common cause of hypo-PP. That mutation has a subtle but very easily detectable problem in the release of calcium. So because we're still struggling to understand the biology of permanent muscle weakness, the idea, and it's still a hypothesis now, is, well, here, one mutation has a slightly different downstream effect. One mutation has a higher likelihood of causing permanent muscle weakness. Maybe there's a connection here. So that's a lead we want to follow up. But just like clinical trials of drugs or other interventions to prevent permanent muscle weakness, even doing the animal experiments is difficult, because you have to wait till the mouse is 12 or 18 months old to do and so it just slows down the pace of doing everything. It's technically challenging, but we will try.
Related, aside from the skin biopsy that you mentioned, is there anything else that we, the patients, can do to support your research? Happy to participate.
So what's beyond my skill set, what I'm hoping to get people excited, is I want to work with clinical trialists, clinicians who run neuromuscular clinics, that get the institutional review board approval and help get the support. And then they're going to need to recruit people to be in the trial. In a rare disease, one of the biggest challenges is patient recruitment. So to make you aware of those and to have you be willing to participate will be huge for accelerating the process of having a good randomized controlled trial, for example, to show that these potassium channel openers are effective. So we'll send out a blast email to all the members when that opportunity comes up.
This person writes in, I have a VUS, which I Googled now. It's variant of uncertain significance. Is that right?
Yes.
In RYR1.
Yes.
Would these therapies that have been discussed have the possibility to help these things?
So this raises a whole interesting topic is, what if someone has recurrent episodes of weakness that significantly impair quality of life, not sure exactly what type of periodic paralysis this might be. It doesn't fit any of the classic ones strong enough to make a definitive diagnosis. And now you've got a variant. Not only is it of uncertain significance whether it changes the behavior of the channel, but it's not one of the top three, sodium, calcium, or potassium. And so my answer would be, there are a lot of things you've learned about here, diet, mindfulness approach, that can help many causes of weakness. I'd have to say that because we don't understand why an RYR1 variant would lead to these types of symptoms, we don't really know whether some of these would work. Certainly, I don't think you could pull out the big guns of gene therapy for that yet. I mean, maybe someone will have that because of the risk of malignant hyperthermia. But if these new variants of the potassium channel openers end up having a more favorable adverse effect profile and are safe to take, I think you could try it. But I can't say that we are able to connect the dots of the biology right now with one of these other variants in not one of the classic genes.
Last few there's plenty more, but we'll run out of time. Which test is more accurate, genetic testing or CMAP?
Yeah, great question. So to make sure everyone heard, which is more accurate, genetic testing or CMAP? It's context dependent. So if you see a pathogenic variant on a genetic test, that's the gold standard. Okay, you're done, that's perfect. When gene testing is ambiguous, it's not because of the quality of the test or the DNA read or something. It's because you see a change, but we don't know what the downstream impact of that change is. That's where that uncertainty comes from. The CMAP test is really good for those cases where the genetics are not revealing of a definitive diagnosis. Either there's a variant of unknown significance or no variant at all. There are a couple of challenges with the CMAP test. It's not done routinely, so it'd be a little struggle to find a neuromuscular group who's willing to do it. It ties up the equipment for a full hour, and so not everybody's willing to do it. The sensitivity is about 70%. So if you don't have an abnormality, we're not quite sure what that means. But on the other hand, the silver lining is if the CMAP test is abnormal, that is a decrease in amplitude after exercise, false positives are extremely rare, if at all. So if the CMAP test is abnormal, that can be the first confirmatory objective evidence that you indeed have periodic paralysis, even though we can't find a pathogenic variant in the genome. So in that context, CMAP is really important for those people.
Very good. Does increasing dose of acetazolamide help recovery from persistent weakness? I'm 73.
Great question. So when carbonic anhydrase inhibitors were first explored in the 70s, they were started with the idea of prevention, prophylaxis. And some of the patients who were recruited were greater than 50 years and weren't quite at 100%. And it was surprising to Birch Griggs and his colleagues that those people improved because everyone thought this was the permanent muscle weakness, this is irreversible. So I would say it's certainly worth a trial, absolutely. And we're looking for better markers. So Frank Weber, who used to come, who's retired in Germany, was an expert at using not only structural MRI, but MR spectroscopy to look at water content and swelling and loss of muscle mass, which might be markers that will help us determine whether your weakness is permanent or might have some degree of reversibility. That's still a great approach. It's still in development. But a simple thing that all of you can do is certainly deserve a trial of a carbonic anhydrase inhibitor. There have been examples of people who thought they were going to be in a wheelchair for the rest of their lives that got up and walked after taking a carbonic anhydrase inhibitor. So
It's amazing. There's a couple more, and it'll be obvious why. One's related, one looks international, and then there was another gentleman here. And I'll invite Teresa, I think, is finishing up our day for us. So whoever is closing for us, if you want to come forward. This one's hypo-PP pattern of symptoms, currently undiagnosed, greatly benefited by daily oral potassium of just 10 milliequivalents. My Invitae 6-gene panel came back negative for any gene mutation or VUS. My doctors do not believe there are any other gene panels worth exploring since my symptoms seem to fit PP and not other muscular disorders. Is that correct? Would you recommend any other genetic testing for me to have done?
Yeah. The short answer is no, the Invitae one is comprehensive, and it's a big fishing trip to go off on whole exome sequence or something like that. It's one caveat: if you have hypo-PP-like features and no variant identified, make sure you've had an EKG and that you don't have Anderson-Tawil syndrome. That's a really important thing to have a cardiologist look at. But otherwise, you only need the Invitae test.
After receiving COVID-19 vaccination and also the shingles vaccination, I suffered major episodes of paralysis. Can you speak to the effect of vaccination that vaccinations might have or can have on hypokalemia?
It's a great question, and all neuromuscular specialists were asked to think about this huge vaccination project with COVID. Of course, the thing to worry about is Guillain-Barré syndrome, which is an inflammatory condition that affects the nerves. There's no known direct autoimmune inflammatory component to periodic paralysis, but I will say that the revved up immune system and stress and perhaps fever that goes along with immunization could certainly cause adrenaline cortisol release. It's going to change potassium dynamics. It's completely understandable that your balance may get a little out of whack around the time of a vaccine.
Thank you. And last question, this one's overseas, and I don't think we'll probably see necessarily this person in the morning. Would you recommend a medication if symptoms from periodic paralysis are not terrible enough, but enough to regularly cause weakness? What are the common side effects from medications for hyper-PP?
So for hyper-PP, usually avoiding the triggers is best, and carbohydrates, mindful, watch, type 2 diabetes or metabolic syndrome, but if you're able to tolerate the carbohydrate snacks, those are great. And low dose of loop diuretics are a good way to keep potassium low and relatively benign. And I would just say for everyone, I don't want to give the wrong message, I'm glad you asked that while we cannot connect all the dots for the biology of what leads to permanent muscle weakness, and there's not a clear association between severity of acute attacks and risk of late permanent weakness, please take care of yourself and try to minimize the acute attacks, because all of us know there's a lot of wear and tear on muscle that happens from that. And fortunately, I think it's completely reversible, but it's a big stress on muscle. So do try to take care of yourself. Absolutely.
All right, thank you. Can we get an applause for Dr. Cannon?
If you would like to know more about periodic paralysis, visit periodicparalysis.org. And if you enjoyed this video and want more, hit that like button, subscribe to our channel, and hit that bell so you don't miss any future videos. It really does help spread the word. You can view other videos about periodic paralysis by clicking the thumbnails to the right. If you have questions, just leave a comment below or reach out to us on social media. We'd love to hear from you.